Structure of bacteria. Bacterial cell structure. The simplest bacterial cells have just the most basic requirements for living cells: a plasma membrane, a cytoplasm, ribosomes, and a chromosome, circular DNA, and a nucleoid. Bacterial cells have no internal compartments, but the four essential structures are sufficient to give them all characteristics of living things. Bacteria respond to the external environment, carry out an internal metabolism, reproduce, maintain homeostasis, exchange carbon dioxide and oxygen, and adapt to changing environments. Some bacteria have additional structures as seen in the image below and described in the following slides. Bacterial flagella. Bacteria may have one or more flagella, or singular it's pronounced flagellum, that move in a rotary motion and propel the bacteria forward. Bacterial flagella are composed of thin, protein fibers. Cell wall. Almost all bacteria have a cell wall that provides a rigid framework and helps the bacteria retain its shape even in the presence of osmotic pressure. This ensures that they won't lyse in fresh water. The cell wall is composed of proteoglycan, a complex carbohydrate protein repeating structure. Capsule. Some bacteria have an additional outer layer called the capsule. The capsule is made of polysaccharides and protein fibers and adds another layer of semi-permeability to the bacterial cell. Some capsules are toxic to humans. For example, the capsule of Salmonella causes food poisoning. Pili. Some bacteria have hair-like extensions from the cell called pili, or fimbriae. Pili, singular pilus, have two main functions. Some help anchor the bacterial cell to surfaces. Others act as conduits for the exchange of plasmid DNA between neighboring cells. Bacterial DNA. Bacteria have a single, circular chromosome in an area of the cell called the nucleoid. The average bacterial chromosome has about 4 million base pairs, roughly 1% of the base pairs in a eukaryotic genome. In addition to chromosomal DNA, bacteria contain plasmids. These are small, circular pieces of DNA that replicate separately from the chromosome. While the chromosome may have 3,000 genes, plasmids typically have fewer than 20 genes. Plasmids are important because they can be transferred between bacterial cells by the process of conjugation. This horizontal gene transfer allows bacterial populations to increase genetic diversity and evolve in the absence of sexual reproduction. The animation GIF at the link below shows the process of conjugation. Plasmids also carry antibiotic resistance genes. Antibiotic resistance genes allow bacterial cells to survive when potential hosts produce antibiotics, or poisons, to kill them. Antibiotic resistance among pathogenic bacteria is a major threat to the future of human health. The correct use of antibiotics can cure pathogenic bacterial infections, while the incorrect use of antibiotics will allow bacteria to acquire antibiotic-resistant genes, become more deadly, and spread. Optional detailed video, click on the link here to see an animation. Antibiotic resistance genes and other bacterial traits may be transferred from one bacterium to another in 3 ways: conjugation, transduction, and transformation. Together, these mechanisms are called horizontal gene transfer. 1. Conjugation. Plasmids are replicated and transferred through pili, bringing antibiotic resistance to new cells. Antibiotic resistance genes may also be transferred by transduction. 2. Transduction. Bacteriophages, viruses that infect bacteria, sometimes pick up bacterial genes in their capsids and transfer genes from one bacterial cell to another when they spread. Antibiotic resistance genes may also be transferred by transformation, the third mechanism of horizontal gene transfer. 3. Transformation. Naked DNA, released from dying cells, is picked up by neighboring bacterial cells and sometimes provides new antibiotic resistance or other traits.